Welcome back! In the previous unit, we explored the context in which industrial biotechnology operates. Let's now look deeper into what industrial biotechnology actually is. As we are about to find out, microbial cells are at the heart of this discipline. In industrial biotechnology, we use microorganisms as miniature chemical factories that convert a substrate, in conventional processes often sugar, into any of a wide and growing range of products. These products range from car fuels to food products and from bioplastics to life-saving drugs. Depending on the combination of substrate and product and on the process conditions, processes may rely on bacteria, yeasts, filamentous fungi, or even algae. Every microbial cell factory, and in fact every living cell, harbors a complex so-called metabolic network. Metabolic networks consist of hundreds of interconnected biochemical reactions that are catalyzed by specific enzymes. Metabolic networks convert simple molecules, such as glucose and ammonium, into all the complex molecules that make up living cells, but they also support formation of industrially relevant products. In yellow, this diagram shows how conversion of glucose into ethanol, currently the largest volume product of industrial biotechnology, is integrated into the metabolic network of a yeast cell. The composition and activity of metabolic networks is not constant, but regulated by the microorganisms in response to external conditions. All microorganisms used in industrial biotechnology today have a very long evolutionary history in nature. For example, the Baker's yeast that we saw on the previous slide naturally occurs in oak trees. In industrial biotechnology, we grow the same microorganisms such as yeast, in large stainless steel bioreactors under conditions that dramatically differ from their natural environment. Moreover, we define performance indicators that are completely different from those active in natural selection and evolution. The three most important performance indicators for microorganisms in industry are captured in the three-letter acronym TRI, which stands for titer, rate, and yield. Titer specifies the concentration of the product, rate specifies the velocity of product formation, and yield defines the amount of generated product per amount of consumed substrate. So, how can we optimize processes to optimize titer, rate, and yield? The answer to this question is twofold. For any microbial cell factory, we have to optimize the conditions in industrial bioreactors. For example, by tuning the temperature, the pH and the oxygen concentration to achieve the economically best combination of titer, rate and yield. Secondly, we very often need to improve the performance of the microorganism in the full realization that this may change optimal process conditions. In practice, optimization of microbial cell factories and the processes in which they operate should therefore be integrated. We need cell factories that perform well under conditions that are compatible and feasible in industrial processes. In both cases, science and engineering go hand in hand. To understand how we can improve the performance of microbial cell factories for industrial application, we need to dig one level deeper in their organization. The genome, that is, the DNA of these living cells, specifies which enzyme and transport reactions are possible in a given cell, and how presence of individual reactions is regulated by external conditions. So if we want to change the performance of a microbial cell factory under any given set of process conditions, we need to change its genome. Using the amazing, still growing toolbox of synthetic biology, we can now edit the genomes of living cells almost at will and implement ever more complex designs for modified metabolic networks. When applied in the context of industrial processes, this editing is called metabolic engineering and encompasses the addition, removal and/or modification of enzymes and transport reactions in metabolic networks. Metabolic engineering has not only enabled optimization of tighter rates and yields of existing processes in industrial biotechnology, but also the production of entirely novel products by microbial cell factories. This slide shows a spectacular example of how metabolic engineering has been used to extend the product range of a microbial cell factory. By adding a series of plant genes and simultaneously introducing changes in the native yeast metabolic network, colleagues at Stanford University in the USA have enabled this yeast to convert glucose into opioids that are normally only produced in poppy plants. The bioreactors in which we grow microorganisms in industrial biotechnology often have a volume of hundreds of cubic meters. This scale imposes constraints on parameters such as transfer of gases into and out of the culture liquid, transfer of heat, and mixing behavior. In this course, you will learn how these constraints can be quantified to support process design and optimization. In addition, you will learn which control options are available to tune process conditions to enable optimal performance of microbial cell factories. As mentioned earlier, the performance of microbial cell factories depends on process conditions. The success of microbial strain improvement, for example by metabolic engineering, should therefore ideally be evaluated under exactly the same conditions that occur in the large-scale industrial processes. Especially in small-scale, high-throughput analysis systems, in which hundreds of microbial strains can be simultaneously tested, mimicking industrial conditions can be a real challenge, and downscaling of processes requires deep insight into the actual industrial system. In this brief introduction to industrial biotechnology, I've highlighted product formation by microbial cell factories in large-scale bioreactors. In practice, this so-called fermentation process represents only one step in a series of processes, or to use a process engineering term, unit operations. Together, these unit operations convert raw materials into the final product. Efficient and robust integration of these different unit operations may impose additional design criteria that, in the case of bioreactors, go beyond performance of the microbial cell factory. Later in the course, you will be introduced to the important discipline of bioprocess integration. This unit introduced you to some basic concepts in industrial biotechnology. In addition, we saw a first glimpse of how design and optimization of industrial processes require science and engineering at different scales, ranging from minute microorganisms to massive bioreactors, and integrating the optimization of cell factories, process conditions, and different unit operations in factories. These subjects will be explored in much more depth and illustrated with inspiring real-life examples in the remainder of the course. We hope to see you in the next unit, in which my colleague, Professor Patricia Osseweyer, will zoom in on the roles of industrial biotechnology in a bio-based economy.